So today we have the honor of having a person who I know just as Rafa, um, but Professor de la Haba. He's part of a tuna, a group of singers, and I had the honor that he serenaded us at one of the meetings in Salamanca. But he is from the University of Sevilla, from the Department of Microbiology and Parasitology, and he is going to talk about a group of organisms that he's been intimately involved with for a number of years, which is the Halomonadaceae. So we're looking forward to this talk. He is also well known within our microbial or bacterial taxonomy group. He is a member of the ICSP, and he is associate editor of IGSM, and he's also involved with Frontiers. So we're really looking forward to his talk. And what is interesting about this talk is that we're moving from polyphasic approaches to network-based analyses. And I think this is a glimpse onto the future. So we're very happy that you are willing to share with us today. And over to you, Rafa.
Well, first of all, I would like to say that it's a pleasure for me to be one of the speakers in the BISMiS live series. So I would like to thank Stephanus for his kind presentation, also the organizer for inviting me. There are many important researchers in our field who has participated in these seminars. So I'm happy to be one of them, but I'm not sure I will be at the same level than my previous college, but I will try to do my best. So, well, as Fanu explaining, today we'll deliver a talk about the taxonomy of the family Halomonadaceae, focus on the history of the taxonomy from the very beginning up to the most recent analysis we are completing. So, let's go. We'll go through the history, the taxonomy history within this family, from the very early studies using now outdated techniques up to more new approaches based on genome sequencing. So the timeline within the family Halomonadaceae can be summarized in the present slide. This slide will be an outline of my talk. The family was first proposed in 1988 by Frasman and colleagues to accommodate the genera Halomonas and Deleia. Then the genus Halovibrio was also described in the same year, in 1988, and more genera were proposed in the next following years, such as Chromohelobacter or Volcaniella or Cimobacter. Of course, several rearrangements occurred within this time, and one of those was the transfer of Volcaniella, the species Volcaniella irralina to the genus Halomonas in 1995. And the next year, the genera Deleia, Halovibrio, and one of the species of Paracoccus alodenitrificans, was also unified within a single genus Halomonas. Continue with this history. In 1888, the genus Carnimonas was described, and then in 2002 affiliated to the family Halomonadaceae together with the genus Covetia. An important date is 2007, where the minimal standard for describing new taxa within this family were published, and also 3 new genera were proposed: Halotalea, Modici salivacter, and Salinicola. In 2009, 2 more genera were described and included also within this family Halomonadaceae. The genera were Kushneria and Dingigomonas. We will talk later about this paper in 2010 that was published from our group, proposing a new approach to use an alternative gene marker alternative to the 16S ribosomal RNA. And this new proposed marker was the 23S ribosomal RNA gene sequence. In 2012, an alternative approach based on the use of several gene markers, the so-called multilocus sequence analysis, was proposed for this family. Since 2012 until the last year, many other genera have been proposed, such as Larsenimonas, previously published as Larsenia and then reclassified as Larsenimonas, or Pterosacispira, Pistricoccus, and the Phytohalomonad, which it's not yet validly published its genus name, and all of them in some way are related or included within Halomonadaceae. An important year was the 2024, where the genus Halomonad was split in 7 different genera based on a signature approach that we'll see later. More recently, taxonomy opinion in 2025 has placed the genera Aluceus spirillum, Marinospirillum, and Pterosacispira within this family. The last year also, a non-validly published genus name, Cernes, has been also published and might be also regarded as a member of the Halomonadaceae. Well, last but not least, I will dedicate the last section of my talk to explain the studies we are currently doing, particularly the use of network analysis as a way to establish a non-subjective delineation of the genera within this family. Finally, here you can see several genus or genera that are marked in boldface. Those are the genera that I consider that nowadays should be placed into the family Halomonadaceae. But as you maybe know, there are different taxonomic opinions, and other taxonomic opinion may place different genera in that family. This is my current taxonomic opinion on this family. So, starting from the very beginning, the first approach that was used by Franzmann and colleagues to describe the family was the 16R ribosomal RNA cataloging technique. And as a result of this study, they could unify the family and create the family Halomonadaceae to unify the genera Halomonas and Andaleia into a single family. Maybe some of them, the oldest people, maybe they know about this 16S cataloging technique, but basically it consists of extraction of the 16S ribosomal RNA, digestion with of the gene with restriction enzyme, and obtaining several oligonucleotides, normally hexamers or longer sequences. And then those oligonucleotides are separated using two-dimensional electrophoresis. And later, the sequence of this oligonucleotide can be inferred, determined by analyzing the migration pattern of the oligonucleotides in these two-dimensional electrophoresis yields. So as a result, what we obtain is like a catalog of oligonucleotides, and we can determine the presence of absence of the oligonucleotide in the studied species, and construct a matrix, a similarity matrix that can be eventually turned into a dendrogram. So this is the approach that Franzmann and colleagues used at that time to unify and create the genus Halomona, the family Halomonadaceae. Some years later, another approach, the numerical taxonomic approach, was used for researchers in working in the field to describe the genera Halovibrio and Chromohalobacter and Volcanilla. Essentially, this numerical taxonomy approach consists of the analysis of many, I would say dozen or hundreds of phenotypic features, including biochemistry or morphological, nutritional, etc. And with all these results, phenotypic feature, we can construct a present absence matrix, essentially a binary matrix. For example, if one of the phenotypic tests is positive, we can assign this test the value 1, and if the test is negative, we can assign the value 0. And with that, we can construct for all taxa under study, and for all the characters, we can construct this present absence matrix that can be transformed into a distant matrix that eventually can be converted again in a dendrogram. More recently, in the 90s, the 16S ribosomal RNA gene sequence was widely used. Up to this days is also quite used. Now has been replaced by genome sequence, of course, but it's still widely used to describe new taxa within all the domains of the life, and of course also within this family, Halomonadaceae. As you may know, after extraction of bacterial DNA, the 16S gene is normally amplified using universal primers, and after amplification, it's sequenced using Sanger methodology. And since the gene is sometimes too long, it needs to be sequenced, cannot be sequenced in a single round, but you can sequence the genus using partial sequencing, and then finally it's necessary to assemble all the fragments to recover the full 16S sequence. Well, with this partial or almost full 16S sequence from different taxa, we can construct the alignment, and the alignment can be used to calculate a phylogenetic tree. And well, of course, using this approach, many new taxas and reclassification within the Halomonadaceae have been proposed. First of all, in 1995, the genus Volcaniella was transferred to the genus Halomonas, and additionally, also the Chromohalobacter was definitely placed within the family. And these studies were based on the use of the 16S ribosomal RNA sequencing. 1 year later, the same approach was used to place the genera Deleia and Halovibrio within the Halomonadaceae, and also the species Paracoccus halodenitrificans was also transferred to the genus Halomonas. Additionally, the already described genus, Cimobacter, was also placed in the family. I'm trying to explain how different genera were described and then moved to the family to this current taxonomic situation. Once the 16S ribosomal RNA gene sequence became popular, researchers tried to search for signature characteristics that could be found in the 16S ribosomal RNA. These signature characteristics allow the affiliation of the genera to different family, and in this case to the family Halomonadaceae. Essentially, the 16S signature are conserved positions in the 16S gene that are characteristics of a certain group. So that means that positions that are conserved across all taxa under study can be used as a taxonomic feature to circumscribe them to the family Halomonadaceae. So imagine that we have these 4 sequence, and in the 4 column, in the 4 position, we have like a thymidine. So this 4 position, which is equal for all these 4 sequence, can be regarded as a signature characteristics to differentiate all these 4 taxa from other that has not this thymidine nucleotide in this position of the alignment. Following this idea of the signature characteristics, several papers propose the use of this signature and they propose a list of signature for the family Halomonadaceae. So the family has suffered different emendation across the time, and the last one in 2007 define the 18 signatures position that you can see here. Nowadays, the signature are not in use. It has not many sense. Now that we can easily sequence all the full 16S gene, it has no sense to do that. But at that time, it was important to define the signature, to define genera, and assign genera to families. Well, in addition to the 16S ribosomal RNA sequence, an alternative gene, specifically the 23S ribosomal RNA, also called the LAR ribosomal subunit gene, was proposed as an alternative marker to infer phylogeny. The main problem when using this alternative marker was the non-existence of universal primers. So we have to design the primers to get the amplification and sequencing of the gene. And the second big problem was the length of the gene, because it has approximately 3,000 base pair, and at that time it was not easy to sequence these longer fragments. So the first we did was to design the primer, searching for conserved region, and use those primers to amplify the gene. Since the gene was so low, we followed an amplification strategy consisting of amplifying the first half of the gene and then the second half of the gene. These two amplicons were used for Sanger sequencing. The first study published using, in addition to the 16S ribosomal RNA, the large ribosomal subunit to analyze the taxonomy of the family Halomonadaceae was those of David R in 2002. In this study, the final result, the final outcome was that the genera Covetia and Carnimona were affiliated to this family. If we compare the phylogenetic tree of this study based on the 16S and then the 23S genes, we can observe a good match between both trees. According to the 16S ribosomal RNA, we could distinguish two groups within the Halomonas, while the rest of the Halomonas could not be affiliated to any of those groups, neither form a group themselves. On the other hand, the species Halomonas marina fell clearly separate from the rest of the Halomona, and that was transferred later to a new genus, Covetia, within this family. And also the genus Carnimona, which has been previously described in 1998, was affiliated to this family. The three inferred based on the large ribosomal subunit were quite in agreement with the 16S, which validate the use of this large subunit gene as an additional marker for phylogenetic purposes. Here, the two Halomonas group previously identified using the 16S ribosomal RNA were also identified, as well as the placement of Halomonas marina, now Covetia marina, within the family Halomonadaceae. Later, 8 years later, in 2010, following the same strategy, we performed a comparison between the 16S and 23S tree topologies, but in this case, we included all the taxa that at that time had been described and belonging to this family. First of all, we can see that the 23 and 16S phylogenies are quite similar, with minor differences, but in both trees we can distinguish these Halomonas group 1, as previously described in the paper from R. Concerning the Halomonas group 2, they were kind of agreement, but the 16S group the taxa not into a monophyletic branch, but in a polyphyletic group. Other taxonomic conclusion of this work was the formal placement of the genera Kushneria, also Salinicola, Modici salivacera, and Halotalea, previously proposed by other authors within the Halomonadaceae. As I mentioned before in the outline slide, an important milestone in the taxonomy of this family was the publishing in 2007 of the minimal standard for describing new taxa of the family Halomonadaceae. Those minimal standards recommended, they don't obligate, but they recommend the use of polyphasic characterization to describe new taxas. Essentially, the polyphasic characterization consists of the analysis of a comprehensive list of phenotypic features, which includes biochemical, nutritional, morphological, physiological, pH, temperature, salinity condition, etc., together with taxonomic features, essentially polar lipids, fatty acids, and cell lipokinones, as well as some phenotypic feature. As in most of the group, it's important determination of G plus C content and DNA hybridization study. So finally, the minimal standard encourage the use of the phylogenetic method to infer evolutionary relationships within members of the family. Well, in addition to the published minimal standard, the ICSP, Subcommittee on the Taxonomy of the Family Halomonadaceae, to which I belong to, also recommended the use of a multi-locus sequence analysis approach, also so-called MLSA, when describing new member of this family. This MLSA approach consists of the use of alternative genes to the 16S. Although the 16S is an universal gene that is present in all the prokaryotes and can be used to infer phylogeny from this tree, it has an important drawback, limitation, and it's when the gene is used to compare closely related microorganisms, because it has a long evolutionary rate and then a poor resolution. For that reason, we thought that it could be possible, would be important to infer phylogenies using alternative genes with faster evolutionary rate than the 16S ribosomal RNA. And those alternative markers are commonly called the housekeeping genes. Well, which genes can be used as a housekeeping gene? Well, requirements for a gene to be used as a housekeeping marker are shown here on the slide. So first, important that the gene is universally distributed across the taxa under study. It's also essential that the gene is present in a single copy to avoid paralogs problem when aligning the sequence. The gene also should have a strong phylogenetic signal, and of course, it must have more resolution than the 16S ribosomal RNA, and it's preferable that the selected housekeeping gene belong to different operon, which is not the case, for example, when you use 16S and 23S gene, belong to the same operon. And finally, it's also recommended that the gene has an appropriate length or size, not too short to retain enough phylogenetic signal and not too long to facilitate the sequencing process. In the case of the family Halomonadaceae, we tested different housekeeping genes, as you can see here, and I mentioned before. Other genes, like for example, the ATPase or the beta subunit or gyrase, the sigma 70 factor or RNA polymerase, or the protein translocase subunit SEC-A. Other genes that were also tested were different chaperones, the uridylated kinase, the beta subunit of the RNA polymerase, or the genes involved in the synthesis of osmoprotectant compound as the ectoin. So the genes that here are shown in green color were discarded due to amplification on sequence problem, because as I mentioned before, it was not easy to design primers that could be used for all the taxa under study. So finally, only these 6 set of genes were used for further analysis. Concerning the evolutionary rate of the selected genes, SEC-A happened to be the fastest evolving gene, followed by RPOD and the beta unit of the urease, the ATPase, and finally the large and small cysteine ribosomal RNA subunits. So this means that the resolution of this gene is bigger than the resolution of that one, and so on. The phylogenetic inference using these 6 genes individually, not in concatenation, but just individually, showed that the topology of the trees was quite similar across all of them, with the exception of ATPA gene-based tree. In the slide, the arrows indicate disagreement between the cysteine-based phylogeny, which was used as a reference, and those obtaining from the use of other individual marker genes. So with the exception of the ATPA tree, the other single marker phylogenies showed only a small disagreement. But this is expectable, so this can be expected. Why? Because the genes suffer of horizontal or lateral genes transfer, and time is normal that different genes have different evolutionary history and then produce different topologies. It must be noted that among all the single gene trees, the one that generates the best matching topology with that of the cysteine was the GIRV gene. In fact, the GIRV gene was almost equal in topology to that of the cysteine-1, and remarkably, it allowed the recovery of all the species within the Halomonas in a monophyletic branch. So all the genus Halomonas was monophyletic, which did not occur in other topologies recovered using this single gene market. Well, considering that the ATPA-based phylogeny was the most different one, we decided to discard this gene and keep all of the other 5. And since a single gene phylogeny cannot be considered as the true evolutionary history of the family, because as I mentioned, the gene can experience lateral gene transfer events, the MLSA approach does not propose the replacement of cysteine with another alternative market, but the concatenation of several housekeeping genes to produce something called a supermatrix, and then use this supermatrix to generate a concatenation. So, well, I was explaining about the concatenation of several genes. So the first option was to concatenate the 5 selected housekeeping gene to produce a good tree with a good level of resolution. And in order to simplify the use of this MLSA scheme, to be used within the family Halomonadaceae, we try to reduce this number of genes from 5 to 3. Why? Well, the reason was to simplify and to encourage people the use of this strategy. The selected genes were those that demonstrated more similar topologies, and we try to include at least 2 fast evolving genes to provide more resolution. And then finally, we selected 3 genes, the cysteine together with the gyrase B subunit and sigma 70 ribosomal RNA polymerase. With this, we constructed a concatenate tree that was almost equal to the 5 concatenate genes. So for simplicity, we recommend in the minimal standard, as the ICSP subcommittee recommended the use of these 3 alternative genes, not as a single market, but in concatenation when describing new taxa in the family. This MLSA approach has been widely used for some years with novel general description within the family, such as the genus Larsenimona or Pistricoccus, or more recently, not yet validly published genus name Phytohalomonas. Well, until now, I have been trying to talk about the old-fashioned taxonomy, which is based on polyphasic taxonomy, or on the use of single gene-based phylogeny, or trees that were based on a small set of phylogenetic markers. But modern taxonomy, as you may know, can take advantage of the high throughput sequencing devices, which allow complete or draft genome sequencing at a reasonable price. Therefore, we can state that whole genome sequencing is nowadays mandatory, or I would say essential in any taxonomy study. Essentially, the whole genome sequence have been used in prokaryotic taxonomy for two complementary analyses. On the one side, we can use the genome sequence to infer phylogenomic trees, which, contrary to single gene-based tree or MLSA tree, are calculated or inferred using a large number of orthologous genes. And additionally, the genome sequence can be used to estimate the so-called overall genome relatedness indices, often abbreviated or known as OGRIS indexes. So let's start with the phylogenomic strategy applied in this case to the family Halomonadaceae. When more species and genera have been described in this family, the well-defined monophyletic groups obtained with the 16S or MLSA approach that I showed you before were not always conserved. Specifically, when we reconstructed a couple of years ago the 16S based phylogeny for the Halomonadaceae, we found that all the established genera form phylogenetic groups, as you can see here, with the exception of the genus Halomonas, the one marked in light blue, which this genus was scattered across the tree, forming up to 7 different phylogroups. Of course, the genus was not monophyletic. So we wanted to test if a phylogenomic tree constructed using the core orthologous gene shared by all the taxa will produce a more consistent result. So here we go. To infer the phylogenomic tree, we selected only the single copy soft core genes. What I mean by soft core genes, so genes that are shared not by the 100% of all taxa under study, but shared by the 90% of the taxa. The reason is that if we choose strictly core genes, the number of genes used to construct the tree is dramatically reduced, and then it's not interesting. We prefer to keep a reasonable amount of genes to construct a more robust phylogeny. Well, this soft core gene tree provided a good resolution, clear differentiation across all the genera within the Halomonaceae, includes the genus Halomonas, because as you can see, all the Halomonas were clustered together, not into a monophyletic group, because here we also have the Idingimonas genus sharing a branch with one of the phylogroup of Halomonas, but at least all the 8 phylogroup of Halomonas clustered together. At this point, we thought of 2 different options or possibilities. So either we could transfer the genus Idingimonas to Halomonadaceae in order to have a monophyletic group for the genus Halomonas, or alternatively, we could split the genus Halomonas into a certain number of new genera with a maximum of 7, corresponding to the 7 to the already existing genus Halomonas plus the additional 7 identified phylogroups. So the first option, transferring Idingimonas to Halomonas, is more conservative, while the the second option is more risky. You will see what I mean later. In any case, our opinion is that the taxonomy tree was not enough as to make a decision. We needed more evidence supporting one of these two alternative options. Keeping this in mind, we decided to analyze the Ogre indexes for the family Halomonadaceae. These, as you may know, these indexes represent any measure indicating how similar two genome sequences are. There are several Ogre's indexes that can be proposed for taxonomic purposes. Most commonly used for delineate taxonomic species or prokaryotic species are the average nucleotide identity, also known as ANI values, and the digital hybridization, also known as DDH. In addition to these two, there are other Ogres that here are marked in green that has been proposed to delineate species with different cutoff values, as you can see here in the slide. The most important are, as I mentioned, is the INI with a cutoff around 95 and DDH with threshold values around 70%. Also, the Ogre value can be used to delineate genera. Genus delineation can also be accomplished using the Ogre's indexes. In this case, all those that are marked in red has been proposed as indexes useful to delineate genera across prokaryotes with the cutoff value indicated in the slide. Well, so keeping in mind that we can use Ogre indexes to define genera, we decide to use the AAI, so average amino acid identity index, for genera circumscription within the family Halomonadaceae, especially within the genus Halomonas. Remember that we have up to 8 different phylogroups within the genus Halomona that have been previously identified using the phylogenomic tree. And the rationale behind the analysis of the AAI values is that AA values within a phylogroup or within a genus must be higher than the AAA values between phylogroups or between genera. So in the figure, the light blue dots represent intragenus values, whereas the dark dots correspond to intergenera value. So a well-delineated genus would be one where it does not exist overlaps between the light and dark blue dots. So as you can see, in all of the lower panel, there is no overlap, and so that means that all those genera and phylogroups within the Halomonas can be easily differentiated because there is not overlap between intra and intergeneric values. So the issues arise when we analyze the top 4 Halomonas phylogroups. Here we can clearly see that there is an overlap between light and dark dots, which means that according to AA values, these 4 phylogroups should not be split into 4 different genera. As I mentioned, the other 4 phylogroups could be divided because there was not an overlap. In addition to the classic AAA index, we use a variation that could also be used to delineate genera in the prokaryote, which is the average amino acid identity, but calculate using only the core genes. As in the previous result, I will not stop a lot here because the result is similar. So the top 4 phylogroups within the Halomonad, you can see a clear overlap between the dark and light blue colors. The other phylogroups in the Halomonas or genera within the family could be easily delineated using this index. Beyond the core genome tree and the Ogre strategy that can be used to clarify the taxonomy of the genera the family Halomonadaceae, we designed an additional approach. This approach was based on the analysis of the Halomonadaceae pangenome to provide additional evidences supporting or not the split of the genus Halomona into different phylogroups, into the phylogroups of the Halomonas into different new genera. The novel approach consists essentially of the identification of signature genes. So I will clarify. So previously I have talked about the signature characteristics of the cysteine S that are conserved position within the alignment that could be used to delineate gender. But here is something different. Now I'm talking about signature genes, not signature position in the alignment, but signature genes. So signature genes are defined as genes that are present only in a certain phylogroups, so they are exclusive of this group, and they cannot be found in any other group, and they must be present in all the taxa in this group and absent in the other groups. Okay, so when we analyze the pangenome of the family Halomonadaceae and we identify the signature genes that are shown in the slide in dark blue dots here, for example, or there in this side here. So we could see that all genera within the family can be identified by a certain number of signature genes. But what about the Halomonas group? Because with the other genera within the family, we could identify. But what about the Halomonas phylogroup? Well, we were not able to detect any signature genes shared by all the 8 phylogroups. So this discouraged us from the idea to keep those phylogroups within a single genus. On the contrary, signature genes were identified for most of the Halomonas phylogroup, supporting their placement as independent genera. For example, here in this group, we can see a certain number of signature genes also here, but the most important is that no signature genes were detected for the 2 phylogroups in the top of the panel. Surprisingly and unexpectedly, if we merge the species from the 2 groups in the top of the panel into a single phylogroup, we could identify a certain number of signature genes. So this is the reason that led us to propose the split of the genus Halomonas into 7 different genus. 1, 2, 3, 4, 5, 6, and 7 here. All these new proposed genera are actually supported by a number of signature genes that are exclusive of this new proposed genera, and they are absent in any other group. Keeping this in mind, our proposal was the following. So the 2 phylogroups that could not be assigned to any signature gene were retained as the true Halomonas genus, while the other 7 phylogroup were reclassified as a novel genus into the family with different names, and we propose name the name that we use were named after a recognizing microbial taxonomy as Bill Graham, Russ Breland, etc. Additionally, as a result of this study, some species within the Halomonas that clustered together with the type species of Modicis alibacter were transferred to that genus on the basis of the phylogenomic tree, the AAI values, and the large number of signature genes that were common to all of them. In order to check our proposal for reclassification of the genus Halomonas, we constructed a second phylogenomic tree that was not based on the core genome, but on 120 marker genes that are commonly used by GTDBA database. In addition to the type strain, here we also include some representative strain from culture and culture microorganisms, and the obtained phylogeny strongly support the split of the genus Halomona in 7 genus because we could see strong support bootstrap value for all the recover or proposed genera. Well, now I will move forward to talk about the study that the new approach that we are currently doing. Firstly, we noticed that the family Halomonadaceae was closely related to the sister family Oceanospiridaceae. In fact, there are several genera that are equally classified either into the Halomonadaceae or in the Oceanospiridaceae, depending on the taxonomy database that we use. For example, the Burgess Manual considered that all of those genera are outside of the Halomonada, while LPSN includes Helovibrio and Pterasacuspira within the family, and GTDB, for example, include all of them except Helovibrio, and a recent paper published in Molecular Phylogenetic and Evolutionary Journal also proposed the inclusion of all the genera within the Halomonadaceae. So the idea is that we have different taxonomic opinion about certain suspicious, we call them suspicious genera, and given the disagreement across different taxonomic opinion and databases, we decided to include in our study, in addition to the species of the genera from the Halomonadaceae, all the genera currently affiliated to the order Oceanospirales. The inferred phylogenetic tree of this type species of the order Oceanospirale, in this case, was based on 162 single copy core genes, include only type strain. As you can see, here the Halomonadaceae family is closely related to four of the previously mentioned genera: Alospirivibrium, Pterasacuspira, Maniospirillum, and Oceanospirillum, but distantly related to the genus Halovibrio. So by now we can conclude that Halovibrio can never be considered as a member of the Halomonadaceae, at least according to the phylogenomic tree. What is not clear is the placement of these four genera in the middle position between Halomonadaceae and Oceanospiridaceae. An important point to consider before making a decision on these four genera is that a phylogenomic tree should be normalized so that all the taxa within the same taxonomy rank or category can be compared independently of the lineage of the evolutionary rate. So tree normalization can be accomplished using a program called PhyloRank that essentially would use is a base tree and calculate something called the relative evolutionary divergence values, also known as RED values. And in the normalized tree, all taxa belong to the same rank should have similar RED values. So if a taxa in a tree in a given taxonomic rank has a suspicious or conspicuous placement, can be easily identified and corrected. In addition to the inferred and normalized phylogenomic tree, if we want to shed light on the placement of these suspicious genera into or outside of the family Halomonadaceae, we decided to follow an innovative approach that was initially developed by Jorge Valcalvo and Jose Vazquez and was tested in the order Mycobacteriales. The idea essentially is to use the Ogre matrix. Specifically, we have used the average amino acid identity matrix to construct a network that will help us to visualize the relationship among the studied taxa. In this network, each node or each dot represents a taxa, and the edge would indicate a relationship between taxa. So that is an Ogre value between this taxa. When an Ogre value between two taxa is about the cutoff limit established, it will be a link or an edge joining both nodes. So first, we use this network approach to study the relationship at the family level within the order of Stephanus because our main idea was to try to place these subspecies genera into or outside the family Halomonadaceae. And so since we are studying the genome at the family level, we decided to use a cutoff value of 25%. So this means that genomes that share AAI values above 25% will be connected with an edge in the network, and those that are not connected means that they share less than 25%. We can observe how the traditional, the classic family Halomonadaceae, here in green, forms a tight cluster with the genera Terasakispira, also the Spirillum, Marinospirillum, Helovibrio also strongly connected. Therefore, according to this figure, all these genera might be placed within the Halomonadaceae. On the contrary, the genus Oceanospirillum, which was also closely related taxa according to the phylogenetic tree, is more distantly related and to the Halomonadaceae and should not be included in the family according to this network analysis. If we increase the cutoff value to construct the network, setting the threshold to around 25-24%, we can still observe that the Halomonadaceae and the other three genera form a single cluster, while Helovibrio start to diverge, which agrees in some way with the phylogenetic tree that places this Helovibrio genus far from the family Halomonadaceae, and we have been previously discarded Helovibrio as a member of the family. And regarding the genus Oceanospirillum, it can from this result, it cannot be considered a member of this family because of the far distant position within this network. Therefore, our proposal is that the family Halomonadaceae should include the subspecies genera Terasakispira in red, Pseudospirillum and Marinospirillum, but not Helovibrio or Oceanospirillum. We are close to finish. If we want to analyze the relationship at the genus level, we must construct the network setting the cutoff value at a higher level. So, for example, if we use a 55% identity as a threshold value, we could see that some of the genera within the family Halomonadaceae would be easily differentiated. Even some of them are unconnected to the main network, which means that they are very good delineated. But on the contrary, the genus Halomonas and the new proposed gene group based on the signature gene analysis that I mentioned before form a cluster that cannot be easily separated using this cutoff value. So if we produce, or we generate again the network, increasing the cutoff value, adjusting the value to 65%, with this AAI threshold value, we start to differentiate some of the new proposed genera, like Brillandella, although the other still remains undistinguishable from the genus Halomonas. Increasing again the cutoff to 65% allowed us to clearly separate Brillandella, but there are still some of the new proposed genera that cannot be separated to Halomonas. And keep in mind that the AAA values usually are used to delineate prokaryotic genus in a range from 65 to 75%. Now here we are above this range that normally it's used to delineate prokaryotic genera. So we should not increase more the cutoff value in order to separate the genera included within this big cluster. However, since we are not able to separate, we will try to increase the cutoff value using 85%, and this is the result. So when we use 85%, Brillandella is now clearly separate. We could see some of the new proposed genera also starting to separate, but still there is this Bilgrantia genus that close together to the Halomonas. So at that point, when I saw this result the first time, I started to think that maybe we should have been follow a more conservative strategy when we decided to divide the genus Halomonas into 7 different genus, because according to this network analysis, some of the phylogroup cannot be differentiated. Seeing that Brillandella, maybe Franzmania and these other 2 could be retained as separate genera from Halomonas, but we must keep in mind that we are using a very high cutoff value to get these genera separate from the main node, that main cluster that is formed by the Halomonas and Halomonas-like genera. So what can we do at this point? Well, anyone can do mistakes. I'm not sure if this was a mistake or not, but an option to revert the changing the taxonomic opinion and reclassify the species of the new proposed genera back into the Halomonas is an idea that had been suggested by the HADCOT Committee on Mitigate Changes in Prokaryotic Nomenclature, which is a committee belonging to the ICSP. And this idea consists of the use of the subgenus rank to reverse or to solve the problem of creating multiple genera as a result of splitting a single genera. So first, we need to complete our study and feel to be sure that a reversion of the new created genera is recommended. In this case, maybe we can propose that some, maybe Bilgrantia or some of them, maybe all of the new created genera based on signature genes approach could be lower to the subgenus category. For example, in this way, the genus Bilgrantia will be renamed as Halomonas subgenus Bilgrantia. This is important because database such as NCBI usually retain the last validly published name, which means that in practice, the new proposed genera will continue to be widely used, even in the case that we propose a new taxonomic opinion. So that is why it's important to rename them using the subgenus rank. Well, just to conclude, thank you to all of us that has arrived to this point. So keep in mind these take-home messages. The first one, that the systematic of the family Halomonadaceae is in contact chain with new taxonomic description across year and new taxonomic opinion throughout history. And also mistake can happen, so even for experienced taxonomies. I don't know if I am experienced or not, but mistake can happen. Second, the new technological approaches can shed light on the correct description of genera within this family. And finally, currently, we are using a combination of normalized phylogenetic tree, signature genes, and algorithm network-based analysis that might be used to establish an not subjective delineation or the limitation of the genera and family within the order Species or Oceanospiridalis. Maybe also this strategy can be used for other taxonomic groups. Finally, I would like to thank all the colleagues that across the time have collaborated with me, taxonomically speaking, sorting out the relationship among the members of the Halomonadaceae. And thank you to all of you for your attention.
Thank you very much. A really very nice talk. And there are a few questions in the Q&A, but before we go there, I wanted to just something that came to my that I thought of when looking at hearing your talk. So is, you've taken us back in history, and the history is really about what happened and how we split up the things, etc., and how the pioneers started. So, if I think of the pioneers, I think of Carl Woese. For me, he is really one of those, in a way, a magical figure, but really outstanding figures that really shaped our thought of the evolution. And if you want to know the history and what's happened, somebody's captured that for us, and this is David Quayman in his book The Tangled Tree. So I would encourage all bacterial taxonomists who's not read this book to get a copy, to listen to it, to read it, because it's really, really interesting to hear. It's The Tangled Tree by David Quayman. So, but that's just an observation I want to make before we start with the questions, because you started at that point where we can really um, see how things evolved and thank you very much for that overview. But let's get back to the questions. So the first is a comment that was made by Aaron, and he says the genus Halomonadaceae was described by Freeland in 1980, and he will be a speaker at the end of November in Mexico at Halifals 2025. And you will be there, Rafa, and therefore they're looking forward to have probably further discussions on this specific topic as well. Then
yes, I will be there.
Yes. So before we get to the more philosophical question of Preben, I want to go to another comment of Aaron, which says some members of the Halomonadaceae family are alkylophilic. Do the alkylophiles cluster together, or are they spread all over the tree?
Well, as far as I know, we could not find any differentiation between the genera based on neither alkalophilic or affinity for salinity, so salinity concentration. The only one is Cimobacter, which is a non-halophile genus, but also this genus is within the cluster with other halophilic genera. So we could not see at this time that salinity or pH can be a characteristic to differentiate the genera into the phylogenetic tree.
Okay, thank you. Then another comment of Aaron, which I think is very valuable. He says the rank of subgenus has been included in the prokaryotic code for many decades, but thus far very little used. In recent months, we saw revival of the subgenus concept due to the paper that you refer to. It may be an interesting idea to devote a future BISMiS live presentation to the question of whether the creation of subgenera is desirable. And I fully agree with that. In my mind, the one thing that we should always keep in mind when we look at genera, because it is a man-made thing, is that we need to take biology and ecology also into account. But thank you for that suggestion, Aaron. We will certainly keep it in mind for planning for next year. I will also speak to Kamlesh. Then there's a question by Siva Kumar asking, how effective is the pangenome approach in identifying species?
Well, the pangenome cannot be used for a single taxa. So you always need to use the pangenome within a group. So you cannot identify a species. So you can just assign species into a group using the pangenomic, the signature approach, I mean. So you cannot use this. In fact, in the figure I showed, you could not see the single species genera. So it's genera that are based on a single species cannot be included in the analysis because all the unique genomes. I mean, this you need a group to analyze the signature gene. So don't use, or at least I don't know how to use the pangenome, not the pangenome. I mean the signature genes to identify species. I don't know if this answered the question.
Yeah, if they want to follow up, the question can, again, another question can be posed. And then let's get to the more philosophical one. Preben Nielsen says, thank you for a great presentation. It was nice to hear your historical chronological summary of the path to where we are with the Halomonadaceae today. Can you put a few words on your hope for the future of bacterial systematics? With GDTB, the MLST analysis become automated using the core genome, which is kind of a extrapolation of your MLST. What is your perception of bacterial systematics becoming digitalized and automated? From my perception, one concern is loss of the physical deposited type strain, which is important for researchers who need reference strains, but there may be other constraints.
Well, this is not an easy question to reply, of course, but well, I agree. At some point, I agree, and I always defend in my presentation. I didn't say here that phenotypic characteristics should not be avoided. So, and in fact, the network analysis to establish non-subjective genus delineation also take into account these phenotypic features. So, we will try to recover the use of the subgenus category will be used also in the case that we need to recover the pre-genomic taxa. So imagine that we are splitting some genera because of the use of whole genome sequence. We are splitting a genus in small new genera. So if we want to go back to the pre-genomic taxa or rank, we should also take into account the phenotypic features. And this is something that we will, for sure, we take in mind when we will redefine, if necessary, the genera within the Halomonadaceae. So again, I will say that the type, not only the type strain, but also the other alternative and culture or reference strain are important, because from my experience, when I construct a phylogenetic tree only using only type strain, I can miss some important reference trait that can affect to the topology of the tree. So don't think only in type strain, but also in alternative non-type strains. And one more thing, answering to Aaron's question, I forgot to mention that I think it's in the slide, the paper, there is already a paper in IGSN talking about the subgenus used for delineation of the, I think it's Mycobacteriales.
Thank you so much. Are there any of the panelists that want to sort of come in here? No. Not at the moment. So again, thank you very much for the time and taking us through this history. I think it was really enjoyable. And as Aaron said, we need to think about the whole issue of creating subgenera. I constantly in debate with my fungal colleagues on the mycologists, because they, in something like Aspergillus or Penicillium, they have subgenus and series, etc., etc., etc. But those are all groups just to help us make it, and it's just whether the name makes sense or not. So I think that debate that would be quite interesting, because we need to decide as a community also if we want to go down a route like that. And I think it is because people have a certain meaning linked to a specific genus name that that you have that, and people don't want to lose that meaning. But Rafael, thank you very much for your time. And for everybody that attended, thank you very much for joining us. And we will let you know what the last session, the 50th session next month, will be soon. So good luck and enjoy the rest of your afternoon or morning or whatever you have at the moment. So, but also, Rafael, thank you for getting up early and giving us this talk.
The pleasure was mine. Thank you for listening. See you soon, dear
See you.
friends.
Well,
Take
Bye.
care.
Yeah.
bye.
See you, everyone.